Before we begin, I'd like to introduce you to Botany Talk Series. Actually, this is a regular agenda of Research Center for Applied Botany, where each of research group will invite an expert to share their insight and current research. And today's Botany Talk, hosted by the research group for molecular plant ecophysiology. So our topic today is about applying molecular approaches in plant physiological research. And today's we are already invited two distinguished speakers from two different institutions. The first one, Professor Emeritus Toshiaki Umezawa. Good afternoon, Professor. And also the second, good afternoon.
Yeah.
Very nice to meet you.
Yeah, nice to meet you.
And the second one, Dr. Robert Caine from Sheffield University. Is it Dr. Robert Caine is already here? Oh, good morning, Dr. Robert Caine.
Hi, can you hear me okay?
Yeah, can you hear me?
Yes, I can. Nice to meet you.
Okay, nice, very nice to meet you. Okay. The two speakers already here. And before we begin, I'd like to invite the head of Research Center for Applied Botany, Pak Imam, to deliver welcoming speech before we begin the presentation. Pak Imam is already here. Yeah. Pak Imam, the screen is yours.
Suara saya terdengar, Mbak Febri?
Iya, terdengar, Pak.
Oke, thank you. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, and also maybe good morning for Robert in UK. So, ladies and gentlemen, and all participants, thank you, and welcome to our Botany Talk Series 14, which is now is organized by the research group for molecular plant ecophysiology. It is a great pleasure for us to have Professor Toshiaki Umezawa from the Department of Kyoto University, Japan. Nice to meet you, Professor Toshiaki. And also we have Dr. Robert Caine from University of Sheffield, UK. Nice to meet you, Robert. And delegate to have you all speakers and also participants here, as we know that one of our scope in our research center is plant biology. So the topic about applying molecular approach in plant physiology research becomes important knowledge. So, ladies and gentlemen, to get a better understand how plant function at molecular level, so recent advance in molecular biology have provided us with powerful tools to study plant physiology in more detail and also deeper. For example, from the gene expression to the protein function, molecular approach are helping us to cover the complex mechanism that control the plant growth or response, and also maybe the plant adaptation. So today we are fortunate to be joined by two experts in that field, because they are willing to share their research experience with us, so we can get more information or update our knowledge related to the fundamental molecular technique used in the plant physiological research, and how the molecular tools are helping us gain a deeper insight into the plant biology. I think they will also share the latest application of molecular biology in understanding how plants respond to the environmental stress. So we can apply this knowledge and also information to improve our research and also develop our research collaboration. So we can also develop the research collaboration with the Kyoto University and also University of Sheffield. I think it will be very nice presentation and then discussion. So I encourage all of you to actively engage in this botany talk. And I hope this session not only provide valuable knowledge, but also get a new idea or a research topic about the potential of a molecular approach in advancing plant physiology. I think once again, thank you for Professor Toshiaki Umezawa and also Dr. Robert Caine, and also research group of molecular plant ecophysiology for organizing the Botany Talk. I hope you all productive and get inspiring session from the Botany Talk series. I think this is a little bit from me. Thank you, and I will give back to Febrina.
Thank you, Pak Imam, for remarkable speech. And before we hear the presentation from our speakers, I would like to, we will have a documentation session, but it's already 117,000, 117 participants here. It will take several screens to be captured. I invite all participants to open the camera, but still muted your video because our team will take several pictures. Please give your best pose and smile. So I will guide you. If I say finish, you can stop, smile, and pose. First screen. Second screen. Third. Sorry. Okay, fourth. And the last one. Okay, finish. Okay, you can still open your camera, but mute your video because in a moment we will hear the speaker, first speaker. And I will share the wait a minute. So we will have 45-minute presentation and it will be continuous after the Professor Umezawa have presentation 45 minutes and then continue to Dr. Robert Caine. And after that, in 20 minutes later, we will have a discussion session. And I'll invite all the participants to actively engage, ask questions or comments because our team already provide free road prize for active participants. So I will read the first speaker's resume. Professor Emeritus Toshiaki Umezawa, educational background, professor finished all the undergraduate degree and also PhD from Kyoto University. And actually, professor has a lot of experience, but this is just, I put just two. From 2023 and up to present, professor has a professor emeritus in Kyoto University and also specially appointed professor at laboratory of metabolic science of forest plants and microorganism research institute for sustainable humanosphere Kyoto University. And professor also actively engaged in professional society and also has several awards. For example, in 2001, awarded the Japanese Society for Plant Biotechnology Academic Award for the work entitled Lignin Metabolic Engineering of Grass Biomass Plants. And in 2024, awarded the Lignin Society Awards for the work entitled Biosynthesis and Biodegradation of Lignin and Related Compounds. There are several research interests of Professor Umezawa. The first one about biosynthesis and metabolic engineering of lignins, bioorganic chemistry and molecular biology of biosynthesis of lignans, non-lignans, and related phenyl propanoid compounds, and molecular breeding of grass biomass plants, which are adaptable to sustainable society, and biotechnological production of anti-tumor lignans, and the last one, evolution of phenyl propanoid biosynthesis. So that's it. Professor, okay. Okay. I will unshare my screen. And Professor, you have 45 minutes to give a talk. You can share your screen.
Yeah.
Oh, I will stop myself. Okay. No. How to stop? Okay.
Okay. Can you hear me?
Yes, we can hear you clearly.
Okay, so selamat siang.
Selamat siang.
Selamat siang, and terima kasih atas perkenalannya. Nama saya Toshiaki Umezawa, Professor Emeritus di Universitas Kyoto. So now please let me speak in English. So thank you very much for your very kind attention. And first of all, I'd like to thank the organizers, especially Dr. Reza Ramadan Ribai, for giving me the opportunity to give this presentation in this special meeting. It is indeed my great honor. So today I would like to talk about the lignin metabolic engineering for sustainable production and valorization of grass biomass. Okay, now please let me begin with the background. Of course, everybody knows that we are now moving from the fossil energy resource-dependent society to the renewable energy resource-dependent society. And this trend has been accelerated by a number of theories and international frameworks such as Social Common Capital, Paris Agreement, Sustainable Development Goals, so-called SDGs, and so forth. And in addition, the COVID-19 pandemic also accelerated this trend. And among various renewable energy resources, biomass is, of course, important for the establishment of the sustainable society. Of course, it is needless to say, however, here we also need to think about the place of the biomass in the society, the future society compared to other resources. For example, how much can be produced and where can they be cultivated, and how about the relation to environmental conservation. So in this regard, what I'd like to explain with this slide is the global material extraction by 4 material categories, as you can see here. Among them, biomass is the annual extraction of biomass is estimated to be about 18 billion tons in 2010, which is much larger than that of the fossil fuels. And in the future, the fossil fuels must be replaced with the sustainable resources such as solar, wind, and biomass. And in the next slide, I'd like to explain the breakdown of biomass, as you can see here. Again, the total amount of biomass is estimated to be about 18 billion tons. And here, the lignocellulose biomass is shown here with the oval background, and such as the crop residues, and it is mainly composed of crop, grass straws, stems or straws, and it is estimated to be about 3.2 billion tons. And as for wood and timber, 1.4 and 1.1 billion tons respectively, and in total amount of lignocellulose biomass is estimated to be about 5.6 billion tons. And this figure is in good accordance with the another statistical data, which I will explain later on. And this slide show the world primary energy consumption. And as you can see here, the burnable renewable energy, mainly biomass fuel, accounts for about 10% of the total primary energy consumption. And so-called fossil fuels, such as coal, oil, and natural gas, account for about 30 and 20%. So in total, they are 80%, 80%. And in future, they must be replaced with the renewable resources, including biomass. And this slide shows the world oil consumption by subcategories. And of course, as you know, oil is mostly exploited for energy production, and they must be replaced with the renewable resources, including biomass. As for petrochemicals, they are made mainly from naphtha, which should be replaced with biomass in the future. So in this context, biomass is very important. And this slide shows the definition and classification of biomass. And biomass can be classified further into three subcategories, such as lignocellulose biomass, non-lignocellulose, and wastes. And lignocellulose biomass is mainly composed of the so-called plant secondary cell wall, and it can be further classified into two categories: tree lignocellulose biomass and non-tree lignocellulose biomass, mainly grasses. And lignocellulose is crucial in the future society because of the characteristics shown in the next slide. In the slide, number one, lignocellulose biomass is environmentally compatible, such as carbon neutral. And second, large amounts of lignocellulose biomass produced and accumulated, and there is no conflict with food supply. And third, lignocellulose production areas are widely distributed, quite different from those of the fossil resources. And number four, lignocellulose biomass can be renewable, of course, under appropriate proper conditions. And number 5 and 6, ability to supply organic or carbon substances and energy. They are very important. Therefore, please let me continue to explain the number 5 and number 6 in the next slide. Okay. So in terms of the material supply in the future, roughly speaking, only lignocellulose biomass can replace naphtha. As for future energy production, of course, needless to say, solar power and wind power are indispensable, very, very important. However, in addition to them, lignocellulose biomass fuel is also essential, as shown in the next slide. Yes, of course, as you know, the output of solar power and wind power are highly variable, as illustrated here in left-hand side. So this is the case of the solar power. But you know, the output of solar power is very, very highly variable. Therefore, output adjustment by thermal power plant is practically indispensable. So this, as shown here in orange. However, here, we cannot increase coal consumption anymore. Therefore, lignocellulose biomass is very important, and it is an immediate alternative to coal. In fact, fuel wood consumption in Japan is increasing monotonically, reaching already more than 20% of total wood consumption. Besides, these biomass resources are new, future, and additional demand. Therefore, we need to think seriously about the areas for biomass production to meet these future demand. And this is this is the current tree lignocellulose biomass consumption, estimated to be about 2 billion tons per year. And this is the non-tree lignocellulose production, mainly grasses, grass straws or something like that, and estimated to be about 3.6 billion tons. By the way, as you can see here in the bottom right, tree lignocellulose biomass from artificial forest is less than half, and most of them are exploited for the wood material and pulp and paper production. So this means that huge amount of tree lignocellulose biomass from natural forest is still being exploited for burning, fuel use. And besides, besides, in addition to fossil fuel use, deforestation also contribute significantly to the carbon dioxide emission. So taken all together, we need to think seriously about the biomass producing area for the to meet future demand. In this context, the potential areas of biomass production is very important, as you can see here. For example, first, areas that are irrigable and potentially capable of growing grass biomass species estimated to be about, say, more than 700 million hectares. And this is the global deforested area during the last 3 decades. This was estimated to be about more than 400 million hectares. And global grassland area in 2022 was estimated to be about 3 billion to 2 billion hectares. And if we cultivate sorghum with 74 sorghum with the production of 74 tons per hectare per year in this area, simple multiplication gives us this figure, 31 billion tons per year of biomass production. Of course, this is merely a very, very simple multiplication, but what I'd like to emphasize here is that there is a large potential of biomass production. And thus, in order to establish sustainable society, we need to think about areas as to where we cultivate biomass plants, species to be cultivated, including their breeding, cultivation methods, and sustainable and efficient utilization of the formed biomass. And in addition, overreaching approach, including all of them is important. And here about areas we are focusing on, we have been focused on the deteriorated grassland formed after excessive exploitation of tropical natural forest resources, and I'll come back to this later on. And in the following slides, please let me explain the species to be cultivated and their breeding, and also improvement of the usability of lignocellulose biomass, which is also closely related to the breeding. And what I'd like to explain with this slide is the lignocellulose biomass plant to be cultivated. We have 2 choices, trees and grasses. Of course, both are indispensable. The right purpose, right species. However, you can see here, at the top of this table, large grasses are very important because of their very high biomass productivity, which are much larger than those of the trees. Therefore, we are focusing on large grasses, especially sorghum. However, as you can see here, at the bottom of this table, lignin content and therefore heating values, grasses are, generally speaking, a little bit smaller than those of the trees. In addition, lignin structures of the grasses are much more complex, generally speaking, much more complex than those of the trees, and ash content are very high. Therefore, they are our breeding targets. Okay. And among the large grasses, as mentioned, we are focusing on sorghum for the reasons outlined here, namely high lignocellulose productivity even among the large grasses, and multiple outputs and drought tolerance, proven cultivation record shown here in the bottom right, and all of which indicate the potential of sorghum to meet future demand. And by the way, as mentioned, lignocellulose usability needs to be improved, and the lignocellulose use can be classified into several categories, as shown here. And among them, we are focusing on chemical component utilization and solid fuel use in relation to their future demand. And chemical component utilization can be further classified into two subcategories: polysaccharide utilization and primary lignin utilization. And in the case of the polysaccharide utilization, generally speaking, of course, there are some exceptions, but generally speaking, the lignin impedes the polysaccharide conversion, such as enzymatic saccharification. Therefore, lignin content reduction or downregulation of lignin biosynthesis should be beneficial. As for primary lignin utilization, in this case, lignin structural complexity is a very, very challenging bottleneck. Therefore, structural simplification of lignin should be important, and of course, augmentation should be beneficial, upregulation. As for solid fuel use, heating values of lignin are much larger than the polysaccharide, and H-type lignin has slightly larger heating values than that of G-type lignin, and S-type lignin the smallest. Therefore, augmentation of total lignin content and H1 G lignin content should be beneficial. And this also leads to the simplification of lignin structure. And furthermore, ash or especially silica content reduction is important in this category. And this also results in the augmentation of lignin content. I'll come back to this later. And moreover, the introduction of so-called easily degradable structures into lignin. Introduction of these structures into lignin is another important strategy. However, in my laboratory, we did not pursue this strategy. So now, please let me explain our lignin metabolic engineering projects, and please let me begin with the upregulation, augmentation of total lignin content. In this case, we took two strategies: overexpression, heterologous expression of transcriptional activator genes, and the other is the knockout of transcriptional repressor genes. And both worked very well, and lignin content was increased largely. Yes, lignin content was increased largely, as you can see here, the raw data. But in addition, we also conducted the in-depth lignin structural analysis, and the result showed that the specific increase of grass characteristic lignin structures in these cases. So I will come back to this later. And by the way, the increased lignin content not only increases heating values, but also enhances stress tolerance. So the augmentation of lignin content is very important. And next, please let me move on to the simplification of lignin structures. This slide shows the proposed lignin biosynthetic pathways and the diversity of lignin monomers and the corresponding lignin structures in various plant lineages. Here, syringil S-type lignin, guaiacil G-type lignin, G lignin, and parahydroxyphenyl H-type lignin. They are so-called canonical lignin units. And as shown here in the bottom of this slide, oasis or grass lignin has the most complex structure because it is composed of the most types of lignin monomers, as you can see here. Therefore, yes, grass lignin is very complex. So therefore, it is crucial to simplify the structure of grass lignin. Please let me continue the explanation of the grass characteristic lignin units. But again, they are so-called canonical monolignals and the corresponding canonical lignin units, and which are common to angiosperms, not only monocot, but all angiosperms. While roughly speaking, very roughly speaking, the yellow ones are lignin units characteristic of grasses, especially gamma paracumulated structure. This structure is characteristic to grass lignin. By the way, Supatomi and others reported the detailed analysis of these parallel pathways leading to the paracumaric acid when she was in our laboratory. And anyway, as mentioned, the gamma position, this position, the terminal position of propyl side chain of grass lignin is highly paracumulated. And in order to simplify the lignin structure, we first try to shut down this shunt leading to S-lignin. So we first carried out the so-called RNA-mediated knockdown experiment of this enzyme gene, CR5H enzyme gene, which is located at this position. And lignin biosynthetic pathway leading to S-lignin. And and we found that as expected, G-lignin content was increased largely. However, S-lignin was still left. But this enzyme gene is not redundant physiologically in rice. Therefore, we next carried out so-called CRISPR-Cas9 mediated knockout experiment in collaboration with the Professor Yuriko and Keishi Osakabe. And as a result, we found that G-lignin content was increased largely. However, unexpectedly, S-lignin was still left. But anyway, we next carried out a similar experiment with the C3'H enzyme gene, which is located at this position, the biosynthetic pathway, in order to shut down the pathway leading to G and S units. At first, we carried out RNA-mediated knockdown experiment, and as expected, H-lignin content was increased largely. However, G and S lignin still left. Again, this enzyme gene is not redundant physiologically in rice. Therefore, we next carried out CRISPR-Cas9 mediated knockout experiment. And as expected, G-lignin was gone. However, unexpectedly, S-lignin was still left. Unexpectedly. And by the way, we also, at the same time, we also carried out the upregulation of this enzyme gene so that we were able to increase synergy lignin content. And at the same time, we also conducted the in-depth lignin structure analysis. And we found that in all cases, PCAS unit content, this structure, the content of this structure was essentially unchanged. So this data strongly suggested the presence of a new pathway leading to this structure, PCAS unit, which is in line with the result of TF transcription factor gene expression manipulation results, which I have just mentioned in the previous slide. Anyway, these data strongly suggested the presence of new pathway leading to these structures. So this means that the metabolic control of the new pathway leading to these structures is necessary for the complete control of aromatic composition of rice lignin. And in this regard, recently, Dr. Lydia Lam and Toima Sensei in our laboratory reported a very important result. So they reported that para-accumulation, this reaction in rice is mediated by redundantly by two PMT enzymes, OSPMT1 and OSPMT2, and their double knockout mutants resulted in the complete loss of the para-accumulated structure. So that means a very drastic simplification of the lignin structure. So this is very important, and this result should be useful for pyramiding the PMT genes and the genes involved in the augmentation of whole lignin and H or G lignin unit content. Now, please let me move on to the another topic, related topic, and aiming at the social realization of our lignin metabolic engineering and the sustainable production and utilization of grass biomass plants. We conducted an international collaboration project together with the Indonesian National Research and Innovation Agency, BRIN, since 2015. This was so-called SATORPS project supported by JICA and JST. And when we started this project in 2015, of course, BRIN used to be Indonesian Institute of Sciences, IPI. And as mentioned, the overarching approach for biomass production, conversion, and utilization is crucial, and this project includes all of them. And in this project, we try to propose a model protocol which would allow us to convert these deteriorated grassland into farmland so that we can obtain the biomass for the pellet and particle board production. And in this project, this project was composed of 3 sub-projects, establishing fertilization method headed by Kobayashi Masaru Sensei from the Plant Nutrition Laboratory of Kyoto University. And the second is the we are in charge of the second sub-project, breeding of the high lignin content biomass. And third is the material development, which was headed by Umezawa Sensei from our institute, Kyoto University. And this slide shows the principal investigators and sub-leaders of this project. And this slide shows the outline of our project. And in this project, Mr. Satoru Midani, the project coordinator from JICA, and Dr. Safendri, Omara Nagamustari, the technical advisor of this project, they helped us a lot, and I really, really appreciate their great contribution to the project. By the way, let me back to my the explanation of our project. And at first, we used rice as a model plant, and we carried out upregulation of lignin biosynthesis and structural simplification of rice lignin, namely augmentation of H or G lignin unit. And on the basis of the result obtained, we next carried out the selective breeding of sorghum with high lignin content. At first, we simply determine the lignin content of 500 sorghum inbred lines supplied from a partner company collaborating company and found that some lines showed very high lignin content, almost equal to those of the coniferous trees. And their trees have already been amplified. And we also carried out mutant screening by tilling technique from 7,000 mutant population of high biomass sorghum on the basis of the gene information obtained in the rice experiment, which I have already explained. And in addition, we also carried out the lignin content-based screening of Indonesian local sorghum accessions and found that some of them showed high lignin content and high G lignin content. This was carried out in collaboration with Dr. Satoya Nugroho's group from BRIM, and the selected accessions were cultivated largely and will be submitted to co-combustion test in coal-fired thermal power plants in Indonesia. And in addition, Dr. Satoya's group submitted some of them to the gamma ray irradiation breeding to obtain high biomass and high lignin content mutant lines, and they amplify the mutants and they will register them in Indonesia as a new cultivar. Now, let me move on to the next silicaless lignin sorghum mutant with higher lignin content. Okay. In our satellite project, we held so-called business matching seminar together with the Indonesian parent companies, and people from the company told me in the meeting that, okay, understood the importance of sorghum biomass. However, its high inorganic content, especially high silica content, need to be reduced for social realization of sorghum pellet. Actually, the inorganic substances can damage the combustion furnaces, and the waste ash, the waste treatment is costly. Among inorganic substances, potassium, sodium, and chlorine can be removed by washout. However, it is impossible to wash off silica. Therefore, reduction of silica content by breeding is necessary. More than 10 years ago, we reported that silica mutants deficient in silica transporters exhibit much less silica content and higher lignin content. Therefore, we thought that the corresponding sorghum silicaless mutant may solve this issue. So we examined lignin content, the wild type grain sorghum BTX623 cultivar cultivated under low silica condition. Actually, this experiment was mainly carried out by Dr. Reza Ramdan Ribai when he was a PhD student in Kobayashi Sensei's laboratory in Kyoto University, and he found a very important result. And as you can see here, he found that, as expected, lignin content was increased significantly. And next, we examined lignin and silica content. The silicaless mutant of the same green sorghum cultivar, kindly provided by Professor Rivka Elbaum. And yes, again, Dr. Reza mainly conducted this experiment. And he found that silica content was reduced drastically and leaf lignin content was increased significantly. However, the lignocellulose productivity of production of this grain sorghum is not so very large. Therefore, we next wanted to have the corresponding silicaless mutant of the biomass sorghum. So, as you can see here, in addition to salicylic acid, toxic germanic acid is also incorporated into sorghum by salicylic acid transporters. Therefore, biomass sorghum mutants that survive under the germanic acid-based screening condition, as illustrated here, expected to be deficient in salicylic acid transporters, and there were silicase mutant. So, and the screening condition illustrated here, very fortunately, we were able to obtain two independent silicase mutant lines, and we next submitted them to the characterization. Yes, we examined lignin and silica content of the silicase mutant of biomass sorghum that's obtained, and the plant was at the early heading stage. And again, Dr. Reza mainly carried out this experiment, and he found that the silica content was reduced drastically and lignin content was increased significantly. And essentially the same result was obtained with the mature plant. And in addition, he found that the so-called HHV value, higher heating value, was increased. He estimated the HHV value, and he found that the value was, increased significantly. And anyway, these mutants can be used as parents for further breeding by pyramiding with lignin content augmenting genes, which can then be offered for social realization. And now let me move on to the next small topic. That is the downregulation of lignin biosynthesis. Well, I will touch this very quickly. Now, as you know, the lignin content reduction is beneficial to the improvement of forage quality, cattle feed quality. And indeed, this strategy has already been commercialized, as you can see here at the bottom of this slide. However, I would say that the application of lignin content reduction to bioethanol production is very challenging, very, very challenging, with the exception of the additional bioethanol production from surplus of the bagasse lignin cellulose in the sugar industry. It does not require the additional expensive capital investment. But anyway, more than almost 20 years ago, we carried out the knockdown of genes encoding the enzymes of the cinnamate monoret pathway, lignin biosynthesis pathway, by RNA technique. At that time, CRISPR-Cas9 genome editing technology was not available, so we employed RNA technique. At that time, our targets were 11 enzymes and 25 genes. So in the next slide, please let me explain the two examples, C-hard OMT and CAD knockdown. This slide, the left-hand side of this slide shows the RNA-mediated knockdown of this enzyme gene, C-hard OMT1. And as you can see here, the gene expression was reduced drastically, which was accompanied by the significant reduction of lignin content and significant increase of enzymatic saccharification efficiency, ESE values, as expected, completely as expected. But at the time, I was thinking that the grass lignin biosynthetic pathways might be different from those of Udico. Therefore, we characterize the kinetic properties of the recombinant enzyme of this enzyme gene. However, the data indicated that this OMT is also located at this step leading to the syringeal lignin, just like eudicot, just like dicot plants. And in addition, the kinetic properties of this enzyme is quite similar to those of the corresponding eudicot OMT. So the result was completely as expected. So, but anyway, this was a fact. So we submitted the result to this journal and published, as you can see here. But recently, not very recently, but later, we found another important, interesting result. And that is the same enzyme, the CROMT, this OMT, was also involved in the biosynthesis of tyrosine, and eventually it involved in the flavonolignin structure, which is the characteristic of grass lignin. And this was carried out as a collaboration with Dr. Clive from the University of Hong Kong. And in our laboratory, this was mainly conducted by Dr. Lydia Lam and Toima Sensei. But anyway, please let me move on to the next small topic, CAD mutant. Well, as you can see here, this mutant exhibit the significant reduction of lignin content, which was accompanied by the significant increase of enzymatic saccharification efficiency. And this mutant also exhibited the so-called brown midrib phenotype, and this is the first report of brown midrib phenotype from C3 grasses, including rice. And at that time, we carefully analyzed the enzymatic saccharification efficiency features of various transformants and mutants of not only the CAD and CROMT, but also other enzymes. And we inferred that the effects of each enzyme on lignocellulose supramolecular structure formation may be different. So, and therefore it was conceived that the reduction of total lignin content, the lignin content reduction would be a useful tool of supramolecular structure studies. So in this regard, we next prepared the single and double knockout mutants of CAD and CROMT and CAD, and submitted the mutant to the characterization, as you can see here. And this work was mainly carried out by Dr. Andre Fadila Martin when he was a PhD student in my laboratory under the direction of Toima Sensei. And okay, they submitted this mutant to the characterization, you can see here. But in short, they found a very nice result. And in short, they found that CROMT, and as you can see here, the bottom right, CROMT and the CAD differently affected the lignocellulose molecular assembly or lignocellulose supramolecular structure formation. So I think this is a very important result, and reduction of the total lignin content using knockout mutants, as explained in the previous slide, is effective for elucidation of lignocellulose supramolecular structure. And the lignocellulose suprastructure, molecular structure is, of course, as you know, is a basis for various academic, scientific, and technological fields. So now please let me summarize my talk. So this slide summarizes our rice and sorghum projects, except for those written in here blue. They are not our work. But anyway, in terms of social realization, they are still halfway. However, for example, silicaless mutants are close to social realization. And in our project, we have conducted lignin metabolic engineering to improve their usability for combustion use and for the production of chemicals. This is basically the application-oriented work. However, although I did not go into details, I believe the feedback to basic science is also there. And the last but not least, I'd like to thank many collaborators, workers, and I would also like to thank the funding agencies and instrumental analytical system. And thank you very much indeed for your very kind attention, and terima kasih banyak.
Terima
Thank you very much.
kasih. Terima kasih, Professor Umezawa. Thank you very much for interesting presentation. For all participants, if you have a question, please hold up for a moment because in the 45 minutes, next 45 minutes, we will hear Dr. Caine to deliver a presentation. But if you have a question, you can also type your question in the chat box below. So we will read it after the presentation of Dr. Caine. Thank you very much, Professor Umezawa. You can stop share your screen because
Okay.
please wait for a moment, Professor, because we will have a 20-minute discussion session after next presentation.
Thank you very much. Terima kasih banyak.
Terima kasih banyak. So I will share okay. So the next presentation will be delivered by Dr. Robert Caine. Dr. Robert Caine is a senior postdoctoral researcher at University of Sheffield. He finished his education undergraduate degree and also PhD from University of Sheffield, UK. And he has several research experience. The first senior research associate from 2021 up to present in University of Sheffield, and also Global Challenge Research Fellow from 2019 and up to 2021 in University of Sheffield, and also Research Associate under Professor Julie Gray Lab, 2016 until 2019 in University of Sheffield. Dr. Robert Caine also actively involved in the Professional Society as an editorial advisor in Plants, People, Planet, also review editor in Frontiers in Plant Science, and also School of Bioscience Sustainable Committee member at Sheffield, member of Institution of Sustainable Food at Sheffield, and also working group member of COAST Pan-European Network of Green Deal Agriculture and Forestry Earth Observation Science. Current research interests of Dr. Caine is about improving the water use efficiency and photosynthetic performance of crops under future climates conditions by investigating and adjusting stomatal and root system to maximize yields. Dr. Caine, you have 45 minutes to deliver your presentation. You can share your screen.
Thank you for the introduction. Can you hear me okay?
Yeah, very clear.
Okay, thank you. Is that clear, your screen?
Okay, yeah.
Can you see?
Yeah, we can see. We can see, yeah.
Okay. Right. So yeah, thank you very much for inviting me to give a seminar today. I'll be talking about stomata, and I'll be going through sort of various different elements of stomata. The three sort of main sort of focuses in my talk are around, firstly, stomatal evolution, and then I will move into stomatal biotechnology. And what we know about stomatal evolution can actually help us to understand kind of like how stomata develop and how they function. And by knowing this information, it actually kind of makes us have a stronger understanding, which we then can apply to subjects like stomatal biotechnology, which is important for strengthening our crops and our trees and things for future climates where we're predicted to have much more extreme environments. And then finally, I'll be talking about stomatal sensing. What I mean by that is remote sensing of stomatal kind of performance from sort of more macro scale kind of technology such as drones. Before I go into kind of more details about sort of those latter parts, I want to start with talking about the beginnings of stomata. Stomata are ancient organs within plants. It appears that early on, when plants sort of fairly soon after they moved from the ocean to the land some 480 million years ago, they developed stomata. And in these first plants, which were very sort of low to the ground, they would have needed a way of exchanging gases with the environment without actually like becoming too susceptible to microbes on the floor, and also to prevent desiccation because there wasn't very much water. So it would have been critical from an early stage to be able to regulate gaseous exchange. And when we look at the land plant lineages, we see that the bryophytes, which are non-vascular plants, 2 out of the 3 groupings of those representative, the mosses and the hornworts have stomata and the liverworts don't, which would suggest that the common ancestor probably did, and then perhaps they were lost at some point in one of those lineages. And then when we move into kind of pre-vascular plants and early vascular plants, some of which diverged and then became extinct, there's evidence for stomata on the epidermis of them. And then as we move into sort of vascular plants, extant vascular plants, which includes the lycophytes, the manilophytes, and ferns, gymnosperms, monocots, which includes our cereals, and eudicots, which is the majority of the flowering plants that you'll see. Stomata are ubiquitously patterned on the majority of epidermal surfaces. And the reason that we know that stomata are ancient kind of structures is that there's fossilized evidence of very early kind of sporophytes that have been dated to be over 400 million years old, which probably derived from after the split from when the bryophytes kind of split from the ancestral lineage. So stomata are something that's been around on land plants for a very long time and have been absolutely fundamental in helping plants to shape the world that we live in today. So it's they're an absolutely fundamental plant organ. So in the current kind of climate, stomata are regulated by a control over turgor. So when stomata become inflated, they increase their turgor, and that opens the stomata. And when they deflate, they reduce their turgor. And this is regulated on the cell surfaces and via ion transporters that regulate the osmotic gradient. So when ions flow out, stomata deflate as the water goes with them. And in flowering plants, typically stomata kidney shaped, and in grasses, which is where a lot of our important economic crops are, they are dumbbell shaped, so they're more elongate, and they have these specialized subsidiary cells which are suggested to serve as ion reservoirs, and they're able to enable the grass stomata to open quicker and be more responsive. And when we think about stomata in terms of what they respond to, stomata opening occurs under low CO2 when it's brighter light and there's higher humidity, and to higher temperature to a point. And this enables water and carbon uptake, solute uptake, and plant cooling. Oppositely to this, in closing, they close in response to higher CO2, lower light, low humidity, and drought. And this enables water retention, prevention of solute uptake, for example, if it's a saline environment and they want to restrict the amount of salt that enters the plant via like the transpiration stream. And they also enable boosting of plant defenses. This is an image of rice stomata, or a rice stomate, which has got papillae all covering the pore. And this was some work that I worked alongside Mutiara Pitaloga, who invited me to give this talk today, which was we've identified these covers which seem to be aiding in kind of like gaseous exchanges at prevention of, so improving sort of water use efficiency. So now I want to sort of talk more about sort of why the evolution of stomata is important and how we can use that information to essentially improve our crop performance. So first things first, do all stomata function in the same way? Because that's important for understanding how they've been shaped over the course of time. Are the genes that governed how stomata form the same, so the developmental genes? And if we can understand how they develop and how they pattern, can we, like I say, use this to enhance future crop performance? So firstly, do stomata function the same way? I'm going to talk about some work conducted by a colleague of mine, Dr. Caine, who's now at Kew Gardens, who looked at moss stomatal responses. So this is a moss, Systemitrium patterns, and these are guard. This is a guard cell, and what Dr. Caine did was he subjected the moss sporophyte capsules to ABA, which is a signal which is often associated with drought, and he measured the pore aperture, so how open the pore was dependent on the concentration of the ABA. And what he found was that the moss stomata seemed to respond to ABA concentration. And he also did a similar thing with CO2, where he subjugated moss sporophyte capsules to differing degrees of CO2 concentration, and he found that high CO2 led to a reduction in the pore aperture size. I just want to stop for a second and say that the responsivity of stomata is a controversial kind of area within the literature, and there is some sort of debate as to like what level of response like earlier diverging rampart lineages have. But this is some really good evidence to suggest that moss stomata can respond to stimuli. And from a molecular kind of and enzymatic perspective, this is regulated by a number of different signaling proteins, enzymes within the guard cells. Under normal conditions, stomata will be more open, and this is regulated by PP2CAs, which prevent OST1, which is a KIT kinase, from phosphorylating SLAC1. And because that is not phosphorylated, chlorine ions remain in the guard cell, and that stops the osmotic gradient from changing. However, in the presence of ABA, under stress conditions, this blocks the PP2CA function, which enables OST1 to phosphorylate SLAC1, and that then enables chlorine to be released from the guard cells, and stomata will deflate as a consequence of the water moving out with the ion. And this was an early study on this OST1 shows that, like, under a drought assay, Arabidopsis, which is commonly used plant for molecular work, when you don't have the OST1 gene, they don't. So the plants still have open stomata, and so they're cooler because they're releasing water, even though there should be a drought stimuli. And following on from this, so Dr. Chater wanted to understand whether or not this OST1 gene could actually function, for the moss version, could function in Arabidopsis. And so what he did was he did some complementation assays. And so we have the COL2, which is the control, and the light gray is the 20 micromolar ABA treatment. You can see that ABA causes stomatal area or pore aperture to reduce, whereas in the OST1 mutant, this doesn't occur. However, when Arabidopsis plants have the PPOST1 gene, they express that gene, this can then lead to a renewed responsivity to ABA, which kind of is some strong evidence that suggests that ancient genes in mosses can actually guide stomatal responses to drought in Arabidopsis. And Dr. Chater also created transgenic moss plants where he knocked out this gene, and this response to ABA was lost. So this confirms that it's very much, this OST1 is very much a player in regulating ABA responsiveness across 400 plus million years. So that's a little bit about the kind of functionality of stomata in respect to on the epidermis. Now, in terms of the development, we wanted to also know, the work I sort of started doing, part of my PhD, how the development was regulated, and then can we, like I say, manipulate this development and patterning in the future. So there's 5 transcription factors: speechless, mute, FAMA, which come from one group, and then ICE1 screen and screen 2, which come from another, and they heterodimerize to initiate and facilitate various transitions and divisions in the stomatal developmental lineage, beginning from a protodermal cell to a meristimoid cell, to a meristimoid mother cell, to a meristimoid cell, to a guard mother cell, to a pair of guard cells. And around about this meristimoid cell point is where a lot of the patterning of the epidermis occurs in the Arabidopsis via these different divisions, which ends up with a mature epidermis after the development has played out, which looks a bit like a jigsaw with stomata slotted in. And when we look at the evolutionary relatedness of these genes in different species, we can see what I just want to mention are these two really eminent female researchers in the stomatal field who have been responsible for identifying and characterizing speechless mute FAMA ICE1 screen 2 in stomatal pathway, and what they've done has been fundamental. And some of this work is working with Professor Bergman, and we basically within the moth we can see that there are two genes that are related to speechless mutant FAMA, which cluster more closely with FAMA, PPSMF1 and PPSMF2, and there are 4 orthologs of screen, PP screen 1 to 4, which are related to ICE1 screen 2, Arabidopsis. So I'm only going to talk about PP screen 1 here, but I'm also talk about PPSMF1 and 2. Do these genes function in during moth stomatal development? And here is a mature moth sporophyte, and then we have a fluorescence image and a SEM image and a bright field image. This is a mutant knocked out PPSMF1 moth sporophyte, and we can see that stomata are completely removed from the moth sporophyte capsule. But yeah, the sporophyte will develop and produce spores. This is a PPSMF2 where there is no significant change to stomatal number per capsule. And this is PP screen 1, and there is a complete removal of stomata. So is this ancestral kind of like, or is there an ancestral interaction of PPSMF1 and PP screen 1 which guides these kind of developmental processes? And so to try to identify if this was the case, we undertook some experiments where we got a gene gun and we made some split YFP constructs. So we made the construct with the SMF and the screen fused to either end of a YFP. And so if they came together, we would get a fluorescent signal. So, and what we can see is when we fuse the two genes to the two split ends in either direction, when we put them, when we transform them into onion, which is a clear epidermis, so you get less fluorescence from autofluorescence from chlorophyll, you can see that there is a fluorescent signal, and this was observed in the nuclear region. And we also found that there were interactions using a yeast-2 hybrid assay. It was only in the media where there was both these two genes together that we got the growth medium and the selective medium. So what this tells us is that there was an ancestral heterodimer that existed 420 million years ago, which first enabled plant stomata to sort of be formed on the epidermis of ancient plants. So moving on from there, we know a bit about development. Now we've talked about the kind of the sort of underpinning development in mosses and Arabidopsis. What about how stomata are patterned on the epidermis? And this involves signaling peptides, epidermal patterning factors and epidermal light patterning factors, and CHALF. I won't talk too much about that. But these signaling peptides, they interact with membrane receptors and proteins, which then lead to a signaling cascade of phosphorylation intracellularly, which then acts upon the transcription factors in the nucleus. And the EPF1 and 2, they negatively regulate stomatal development by binding with these receptor proteins and receptors and sending phosphorylation signals that block development, whereas EPFL9 prevents this binding and therefore stops that signaling cascade. And from a amino acid perspective, we can see differences that like signature differences in 15 residues within the terminal region, which kind of show that which of these peptides from a phylogenetic perspective are negative regulators and which are positive regulators. And you can see the pink arrows are the 6 conserved across all, and the 2 with the black arrows are showing the ones that don't have those 7th and 8th cysteine residue, which are only found in the negative regulators, and EPFL9 is a positive regulator. And I just want to mention Professor Julie Gray, who is one of my PhD supervisors and also my boss during my first postdoc, who has been fundamental for EPF research for 15, 20 years, and you know, is a real massive player in this field. So I don't want to talk about this whole phylogenetic tree. I just want to talk about EPFL9 and 10 and EPF1, 2, and this EPFL7 here, although this is more of an anomaly, so let's just focus on EPF1, 2 and EPFL9. I should say EPFL10 is a graph-specific innovation in themes. And so we said that the margins of EPFL9 is and 10 are positive regulators, and EPF1, 2, and as I just said there, 7 is a negative regulator. There's one MOS EPF gene, PPEPF1, that clusters with the negative regulators, and it has the same signature of cysteine residues, despite, you know, the ancestor of these plants being diverging 400 million years ago. And that enables this kind of like loop formation. So what happens when you knock out this gene in moss? Well, here's 2 fluorescent images, one being the wild type with sort of the little light colored stomata sort of fairly regularly patterned, whereas in the EPF1 knockout, you can see like there's chains of light that are kind of signifying clustered stomata. So this is saying that this gene normally negatively regulates stomatal development and patterning, and when you knock it out, you get these cluster lines. And this is evident when we do a stomatal count. We see that you get approximately twice as many stomata per capsule. And I created an overexpression construct to validate this, and the opposite sort of phenotype occurred, where we have less than half of the normal number of stomata. So this EPF gene seems to function, well, it functions in Arabidopsis and it functions in moss, so there's kind of a high probability that it also functions in crops and other plant species. Just before I go on to the biotechnology angle, I just want to show you some information about stomata in respect to their function in mosses. So we undertook an experiment where we wanted to assess how quickly the sporophyte matures in the wild type, and then in plants that had no stomata on their sporophyte. And what we found was that like the wild type plants matured faster than the ones without stomata, which kind of suggests that they may have been aiding in dehiscence and or carbon capture. So dehiscence is a drying out and release of spores, so they may have been aiding in drying out. And these are some images of the underneath of sporophyte capsule conductive grown on these kind of plate medium where it's very dry, and you can see that the stomata are kind of you have these black markings where there's a dry down and a hollowing of the stomatal canopy pores. But you don't get this in the mutant lines because stomata don't form, neither do the underlying kind of pores. And some more interesting and more recent work is presented here, where we see that when you have too many stomata in a wet condition, you get this sort of blackening on the epidermis in the PPF1, not in all plants, but in some. And this kind of shows that the integrity of the epidermis has been lost. And so, on the one hand, you know, mosses need these stomata to increase their fitness by dehiscing faster. But on the other hand, if you don't have the patterning correctly on the epidermis, then you may be vulnerable to attack. So, moving into stomatal biotechnology now, moving across to rice, there's going to be an EPF theme to this, as you might expect. Rice obviously is hugely important to the globe and provides 20% of daily calorific intake for over half the world's population. However, to produce 1 kilo of rice, it takes around 2,500 litres of water. Particularly for irrigated rice, paddy rice, it's very water intensive to cultivate. And also it results in a lot of methane emissions, which is not good for global warming. So we need rice varieties that use little to no additional irrigation, so they're predominantly rain-fed, which are capable of withstanding multiple abiotic stresses. And these stresses include severe drought, rising water salinity, and/or rising temperature. And there are, of course, others as well, but I will be discussing these. So I talked about the MOS EPF, PPEPF1. There are two negative regulators, or what may be negative regulators of stomata development, that cluster in the same clade as PPEPF1 and the Arabidopsis. And these have the same cysteine residues. So they both would be potentially good candidates to look at for reducing stomatal number or density, which is the number per unit area. So we've been looking at OSEPF1 and three challenges for developing climate-ready rice, by reducing stomatal density. One, can we first, is it possible to reduce stomatal density? And I'll talk about that first. Will this make rice more resilient to water, nutrient, and/or heat stress? And finally, is there scope to find natural variation in stomatal size and stomatal density, which might be useful for GM, that when GM techniques aren't possible? So if there is societal kind of negativity towards like genetic methods. So let's start with the EPF reduction of stomatal density. Here's some confocal images of two transgenic lines with reductions in stomatal density caused by overexpression of OSEPF1. We've got the W and the F lines, and the yellow atrophies indicate where stomatal development has been inhibited in the mature epidermis. And when we quantify the stomata, we can see that in the wild type, the black, most of the cells that we see as in the stomatal lineage are stomata, whereas for the OSEPF1 W and S lines, we see much more of this guard mother cell formation, and to a lesser degree, some post-GMC. So cysteine works during the stomatal lineage ordinarily to regulate stomatal development, and when you overexpress it, it inhibits this. And this is also evident in the stomatal index, which is the number of stomata relative to the total number of cells on the epidermis. And when we look at gas exchange using infrared gas analysis, we can see that when you have a large reduction in stomatal density, like for example in this OSEPF1F line, you get a significant reduction in photosynthesis and a significant reduction in stomatal conductance. However, if there's only a moderate reduction, then you do not see these significant differences. And when you look at the overall water usage, we found that both transgenic lines showed actually a similar reduction in the amount of water use from days 28 to 35 when we measured water use. So that kind of says, yes, we can reduce stomatal density, and we can have some level of control about how much we can reduce it. But will these changes make rice more resilient to water, nutrient, and/or heat stress? So this is a thermal image with the darker blue being cooler, which is implying more stomatal water release, and the lighter blue being warmer, implying less stomatal water release. And this is under well-watered conditions. And then when we do a 3-day drought, we see a reversal of this water flux, because the plants on the left-hand side have used all the water, whereas the middle and right OSEPF1 overexpresses haven't. And when we quantified yield, we found that a moderate reduction in stomata actually increased yield during drought, whereas the S lines we saw a similar response, possibly because of the lack of carbon acquisition. And I should say, under normal conditions, we don't see any differences in yield between the transgenic plants and the control plants. So next, I'm going to talk about salinity. We did an experiment 1 week in water, and then 4 weeks applied with 20 millimolar of salt. And after 2 weeks in the salt, at week 3, we did some gas exchange analysis, and we see that the control plant had reduced stomatal conductance, and this sort of brought down photosynthesis to a point. And in the following week, we then measured the kind of breakdown associated with increased salt pathology, and looked at 5PS2, which is a photosystem 2 kind of, that's a measurement of the fluorescence capacity of the photosystem. And we found that when we had this salt treatment, that the photosystem 2 was losing its capacity to fluoresce, which is clear from the pathology. And by week 5, we wanted to understand whether or not there was a difference in the salt buildup, and we found that using ICP-OEF, which is inductively coupled plasma optical emission spectroscopy, when we measured sodium content, we saw saw that there was less of a build-up of sodium, which kind of supports the idea that less sodium is being taken up because there's less stomata that's pulling it up from the roots. So moving now from salt to heat. On the left here at the top, we have stomatal density counts, just to show that there is a response of stomatal density in the control lines, but not in the transgenic plants when we move from 30 to 35 to 40 degrees. And when we look at pores, which is only looking at the data from 30 to 40, we see that there's a gradual increase in pore area for 30 and a much more dramatic increase when we get to 40 degrees, and this is evident here. And when we look at the photosynthetic responses, we can see that this kind of increases the stomatal conductance, particularly in the OSEPF1S line. And to a certain degree, we see that the reduction in photosynthesis is actually lost, and they become more similar at that higher temperature because more gas exchange is occurring as the stomata in the transgenic lines are increasingly open. So this is sort of showing there's good evidence that reducing stomatal density can really improve abiotic stress resilience. But like I said, that there may be societal kind of objections to such techniques. So we also wanted to see whether or not we could mimic and find sort of like natural varieties that might have similar capacity as our transgenic lines. So we grew 72 natural varieties of land races and cultivars, and compared the stomatal and physiological performances. So you can see here that we've picked these coloured ones, and then the green ones are the transgenic parts. So red equals like large stomata, reduced stomatal density, and blue equals higher stomatal density and reduced size. And the first question I wanted to ask is, how do these plants compare when grown under well-watered conditions? So these are the plants that we pulled out. We measured the stomatal density again on the plants that we surveyed and the stomatal size. And the stomatal size doesn't follow quite as much of a nice trend. It seems like there's some level of kind of breakdown between these two attributes in the transgenic line. And this is evident when we look at the regression analysis of just the regressions only covering the natural variety. So you can see that the EPF overexpressor lines don't follow the same trend necessarily. And that's kind of what I've been alluding to there. So this, I want to talk a little bit about anatomical, abaxial anatomical GS-Max, or more broadly anatomical GS-Max. I don't want to talk too much about all of the formula that goes into this. I just want to say that there is suggestions that if you've got a higher GS-Max, then you potentially can have a more speedier stomatal responsiveness, which might enable water saving capacity or capabilities when there's fluctuations in light or there's a drought signal or low humidity in the air. So we calculated abaxial anatomical GS-Max and found that high density small stomata, these ones here, had the highest GS-Max, whereas the transgenic plants had the lowest GS-Max. And when we measured stomatal conductance using a barometer, we found that, broadly speaking, like higher stomatal density and smaller size led to higher stomatal conductance, although there was one outlier in the UCP 188 line, which had a very high stomatal conductance. And this kind of just shows the relationship, not including the transgenic lines, between the SD and the leaf GS-Max and the SD and the leaf GSW, which is confirming those relationships. So OSCCF1 plant has the lowest GS-Max. Natural rice, the land race cultivars with the highest SD, and small SS have the highest GS-Max. This suggests that these varieties might be more responsive to certain abiotic stresses. So before I talk about this responsiveness, I just want to kind of align stomatal metrics with biomass or plant size. And we can see that stomatal density minus the transgenic, typically the plants that are larger have a lower stomatal density, and they have a larger size, whereas the plants that are smaller have higher stomatal density and smaller stomatal size. And this is reflected in these regressions. Again, this doesn't include the OSEPF1 plants, which have an unusual kind of relationship. Yeah, so this kind of sets the scene. So there's a link between plant size and stomatal size and density, in that smaller plants have higher stomatal question is, does SF and SC really matter in respect to abiotic stress resilience? We've already kind of outlined there is a kind of a strong relationship with size, and if the plant's bigger, it may have a higher water use. But what we see is that plants with higher stomatal, higher stomatal density, the natural varieties and smaller size, typically have a slightly better drought tolerance than the larger stomatal size, lower stomatal density. But the transgenic plants have the best sort of drought sort of responses of all, maintain the highest 5PS2 values at the tiller base. And this is also reflected 7 days after watering when we look at the amount of newly emerging leaves that occur. And here's some images to sort of show this. This is the transgenic plants, and these are the large plants with large stomata and fewer, often like low stomatal density. And these are the smaller plants with high stomatal density and small stomata. So how about salinity? Well, we did a terminal drought experiment, adding 50 millimolar of salt on day 16 and just growing the plants and just seeing how they responded to the salt. And the quantification at day 41 showed that the EPF plants had the highest 5PS2 values, and that there was no real wrong relationship between sort of, or no definitive relationship between sort of size, stomatal size and density and salinity response. And kind of just clarify there. Now, the other stress that I'd like to talk about is temperature and rising vapor pressure deficit as well. So I said before that temperature opens stomata, but actually vapor pressure deficit reduces stomatal kind of aperture. And you can think of VPD as like a hot, dry air. So it's kind of like a drought in the air for the plants. And the reason why VPD increases with temperature is that it requires to have, as the air gets warmer, you need more water in the air to maintain the same level of relative humidity. So essentially there's this disparity. So VPD, high VPD is more likely to occur at higher temperatures, and so it can then lead to stomatal closure. And this occurs, of course, when there's drought environments, there's not any water in the area. So we undertook an experiment where we looked at temperature and VPD increases. So we started at 32 and went to 42. And we tried to keep the humidity the same, which would mean that the VPD would increase, and it did. But obviously there's limitations to how much water we can add into a system like this. However, we still maintained a very high VPD at 42 degrees. And we first of all wanted to test large stomatal size and low stomatal density versus small stomatal size and high stomatal density. And here's the first part of the assay is the photosynthesis. And what we can see is that the large stomata with fewer low stomatal density, they didn't respond as much with their photosynthesis, whereas the plants that had smaller stomata and lots of them rapidly reduced stomatal photosynthesis, and this obviously increased the rate change. And if we look at stomatal conductance, which guides this, we can see that this is probably caused by the rapid reduction in stomatal conductance. And this is what we were talking about. I was talking about previously about GF max. It suggests that potentially the GF max is linked, which is higher in these high density, small size, high density plants. And here's the response per minute. Because the plants with high density and small stomata close quicker, they have a higher intrinsic water use efficiency, which is kind of good for water saving capacities. On the other hand, what that means is you're releasing less water, and so potentially you're less likely to be able to cool the leaves. Whereas with the large size, low density plants, the transpiration continues because stomata didn't close. And we did the same assay just to compare the difference between control and EPF overexpressing rice lines to see what the responsivity of EPD would be. And what we found was that there was typically a sort of faster response in terms of photosynthesis to a reduction in the control lines relative to especially the OS-EPF1 OEF line. And this was to some degree, looks like it was driven by stomatal conductance, although there wasn't significant differences. And we see that this kind of closure of stomata kind of actually slightly increased, or again, not significantly so, but there's a trend towards an increase in intrinsic water use efficiency. And because stomata didn't close in the transgenic line so quickly, the transpiration rate increased at a faster rate because the water was being pulled from the plants more. So just to conclude on the second part of the talk, OS-EPF1 plants have lower SD than traditional cultivars and landraces. Small SF and higher SD in natural varieties is linked to higher CS max and faster stomatal responsivity. OS-EPF1 plants with very low GS max are most drought tolerant, followed by smaller landraces cultivars with small SF and high SD, which is probably linked with the higher GS max and faster responsivity. Very low SD transgenic plants which have low GS max have increased salt tolerance. And the last experiment suggests there may be a trade-off between high productivity and stomatal responsivity when in future climates where BPD is much higher. So the last part of my talk is about 3D stomatal, or 3D and stomatal sensing. And when I say stomatal sensing, I mean remote sensing of stomatal performance from sort of the macro scale. The reason why it's important to understand plants at macro is we need to obviously develop improved crops and efficient nutrient fertilization strategies if we're going to maximize productivity and minimize waste. And that includes emissions and waste attributed to losses. So this is not about transgenic, but we've created a system here where we can look at differences in physiological performance using nutrient fertilizer differences, which I'll go into more now. Like I say, we can use these remote sensing techniques to look at things over much larger scales, which will enable real-time assessment of stomatal and photosynthetic performance. And I'll talk about both stomata and photosynthesis here. By applying more nitrogen fertilizer, plants can increase chlorophyll biosynthesis, which in turn does lead to higher photosynthesis and also opening of stomata. Sulfur, which is another nutrient that's important for assimilation of nitrogen, helps nitrogen to be pulled up by wheat and enables higher photosynthesis and possibly stomatal gas exchange. And sugar application in the form of sucrose can impact microbial communities, which might impact nitrogen uptake from the soil into plants and therefore also impact gaseous exchanges. So this work is conducted in my current role with Dr. Holly Croft, my boss, who's a Future Leaders Fellow and a lecturer at the University of Sheffield. And this is also mosaic created from drone flights. There's 994 images stitched together, and it covers various different nutrient treatments. And within this study, we wanted to sort of test the application rate, so regular nitrogen versus little and often nitrogen, to see whether application rate had an effect on performance, because it suggested that little and often nitrogen application might reduce the amount of emissions because less nitrogen is lost to the environment. We wanted to test how sulfur application impacted on nitrogen uptake. And we also wanted to see whether if we altered the microbiome by adding sugar, would that impact on the amount of nitrogen uptake, and we can then measure the gaseous exchanges and photosynthesis changes. So first things first, to get a proxy of biomass during growth time, we measured LAI, which is leaf area index, which is the amount of total leaf area per unit ground area. So higher LAI is the higher greater potential for biomass. And then a value of around 5 equals a full canopy. And we can see that the sulfur treatment, in addition to the regular N, led to the highest LAI values, whereas sucrose, if anything, had a negative impact overall. And when we measure leaf chlorophyll using a spectrophotometer, we can see that similar sorts of responses are found with leaf chlorophyll compared to what we saw with LAI. With fertilizer in any form having a drastic increase in chlorophyll, and you can see on this image of the field, the areas with the red where there's no fertilizer being applied. And this is then linked with photosynthesis, which, you know, with high nitrogen you have and high chlorophyll you have high photosynthesis, and then this is linked with sulfur as well. The rate of application of nitrogen doesn't impact chlorophyll or photosynthesis, but sulfur sometimes improves its performance, and sugar seems to not aid performance. The VC max is a measure of maximum rubisco carboxylation, which is a measure of photosynthesis, and this is linked with chlorophyll. So if we know, if we can see this relationship, which we can, leaf chlorophyll is linked with VC max, then we can then see whether or not we can use vegetation images such as the Meris terrestrial chlorophyll index, which can be taken by hyperspectral imaging or drone imaging. And then if we can compute this MTCI value, we can map photosynthesis on the field. And this is what we've done here. So N, S, and sucrose related differences in VC max are captured by hyperspectral ground data and optical drone data. And the vegetation index MTCI can be used to map this performance. So moving quickly on to stomata, I'm not far away now. We can see here that stomata open. You can see this from the impression when you have fertilizer in play. And we see that plants with fertilizer have higher stomatal size and lower stomatal density because the plants are growing bigger and the stomata are stretching out. And this is, you can see there's higher stomatal conductance, which is attributed to the increased photosynthesis. And we use thermal imaging on the ground and showed that plants are cooler, with which reflects the increased gaseous exchange. And when we look at the thermal orthomosaic, and then we measure the temperature on there, and we compare that to the gas exchange with the thermometer, we see a similar response, which suggests that we can use drone imagery to look at differences in gas exchange as well as, so that's the thermal imaging and previously the optical imaging. And just finally, we wanted to see whether or not these optical and thermal data we collected during peak season could be related to yield at the end of the season. We can see again that sulfur is important for maximizing yield, whereas the rate of application doesn't seem to impact, and sugar, if anything, has a detrimental effect. And what we can see, as I've just said, is that fertilizer application didn't impact yield. X was typically beneficial. Sucrose often led to reduced yield, but in the final kind of final results, there was a 0.94 R-squared correlation between MTCI and yield. Drone leaf temperature was 0.62. And we also found that stomatal size was actually a good correlation, showed a good correlation with final yield. Just to summarize, wheat stomatal development and physiology is very dependent on nuclear fertilizer application. Any form of N fertilizer application increases SS and reduces FD. Increased N fertilizer increases photosynthesis, which is linked with GSW also being increased. Optical drone data capture is extremely accurate for predicting chlorophyll and final grain yield in wheat. And thermal drone data is a good proxy for stomatal conductance and yield, and further applications capturing other abiotic stresses, drought, etc., will increase the utility of this emerging platform. And with that, I'd like to say thank you for listening. I hope you've enjoyed the talk, and yeah, I'd just like to acknowledge these people, particularly my former boss, Professor Judy Gray, and my current boss, Dr. Holly Croft. Yeah, any questions you may have in the session to follow.
Thank you, Dr. Caine, for great talk, great presentation. And today we have discussion session for 20 minutes. In the next 20 minutes, please raise your hand. You can use the React emoticons below to raise your hand. There is one question actually in the chat box from Nurfianto Giarpanding Pangidung. Pak Nurfianto, would you mind to open your camera and deliver your question directly, or I just read your question? Yeah. So the question is addressed to Professor Umezawa. The first question, there are three questions. The first question, are there any differences in treatment approaches in lignite metabolic engineering for bioenergy biomass from C3, C4, CAM plants? And the second question, with the reduction of silica content and simplification of lignin, how does it respond to the plant characteristics of tolerance to abiotic and biotic stress? And the third, is there a trade-off between if we engineer plant lignin for bioenergy purposes against the plant character for other uses? For example, in the case of sorghum as a food, for feed and bioenergy crop. The question is addressed to Professor Umezawa.
Yes. Can you hear me?
Yes, we can hear you, Professor.
Yeah, thank you very much. Very good questions. And about the first question, well, of course, I cannot rule out the possibility that there is a difference among C3, C4, and CAM plants. However, the rice, which is the C3, and sorghum and corn, which are C4 plants, they exhibit similar tendency in when we carry out the metabolic engineering, lignin metabolic engineering. So I think that the difference is not so very significant. But of course, there could be a difference. Is that okay? Is that answer for you? Okay. And okay, now let me move on to the second question. With the reduction of silica content and the simplification of lignin, how does it respond to the characteristics of tolerance? Yeah, that's very important. Well, this is a very good question. Basically, these are issues that need to be addressed in the future. So, but regarding the silica content reduction, I think there's no significant reduction in growth when grown under controlled conditions. But Dr. Reza, Pak Reza,
Hi.
can you hear me?
Yes.
I think
Yes,
you conducted the cultivation by yourself. So could you please add your comment on it?
Okay, thank you so much for the questions and the time for me. So related to the second question, it's very nice questions. So yes, as we know that silica and also lignins has a similar function for the plant bodies, but silica and also lignin support the plant for the mechanical properties. When we reduce silica, it's likely compensate the support system of the plant by upregulating or enhancing the lignin contents. That's why when we reduce silica, the lignin content is up. And how to compensate the plant growth and also the tolerance to the abiotic or biotic stresses, it seems that when the mechanical strengths up and enhanced, then the abiotic or biotic stresses can be supported by the lignins. It seems theoretically, but we need to make the evidence for the futures. That's the question, Sensei. Thank you so much.
Terima kasih. And about the third question, is there a trade-off when we engineer the plant lignin for bioenergy purposes against plant such and such? Yeah, they were also very, very good question. Of course, there are many, as you know, that there are many types of sorghum cultivars. Some are grain sorghum, good for the production of the grain. And some are sweet sorghum that are good for the sugar production from the stem syrup. And there are some biomass, so-called what we call biomass sorghum, that are very good for the biomass production or cattle feed production. So that means basically the right purpose, the right species. And as you know, the grain sorghum or sweet sorghum exhibit, generally speaking, less lignocellular biomass productivity. Therefore, biomass sorghum is better for lignocellular biomass use. However, this is it's a kind of higher order decision. But there is also the idea, I think, of using biomass of the stalks or bagasse after harvesting grain and sugar, with grain sorghum or sweet sorghum. Okay, just cultivate the grain sorghum or sweet sorghum and just harvest grain and sugar first. Then we can exploit the stalks. In these cases, evaluation of the total system, for example, using life cycle assessment, LCA analysis, or something like that, should be necessary. So yeah, basically these are the issues that need to be addressed in the future. So I think there are, we need to think about many factors in this case for the social realization. Is that okay?
Yeah, thank you, Professor Umezawa. I think it's clear explanation to this question. I invite a participant to ask a question. Okay, I want to raise a question to Dr. Caine. What if the effect of reducing the stomatal size and also increasing density to the productivity or yield of the rice? And the second question, if we talk about climate change, we also talk the carbon dioxide. Is there any is there any effect of carbon dioxide to the development of the stomata?
So, like Prof. Umezawa, like most of the kind of initial work we've done has been in growth chambers. So it's kind of limited amount of environmental conditions the plant or the rice has been experiencing. But under the conditions that we've grown them and we've measured yield, we've not seen any kind of differences in grain yield in rice. We did see a reduction in wheat when we had a severe reduction in stomatal density, but it seems to be like a gradient effect. So if you have sort of 50%, then in most situations that seems to be okay, and you benefit from improved resilience under abiotic stress, which is obviously beneficial. And I think it comes back to what Prof. Umezawa was saying, is like you have different things for different situations, right? So if you have a situation where you are going to have infinite amounts of water in a rice paddy and you're not worrying about any climate factors or anything, then that's okay. But if you are wanting to move to kind of more of an upland where rice and water is more limited, then reductions in stomatal density may well be hugely beneficial. Could you repeat the second question, please? Early morning, my brain.
Okay, the second question is about carbon dioxide. There for the stomatal development, is there an influence to the developmental of stomata?
So I've not personally looked at rice CO2 responses, but I do know that in Arabidopsis that there is regulation of stomatal development by EPF2. They suggest that EPF2 is seized differently depending on CO2 concentrations, and then this regulates the number of stomata that form, and this essentially suggests a mechanism for reducing stomatal density at higher CO2 concentrations. In terms of heat stress, like we showed there seems to be kind of like a nullification of kind of response to the environment when you play around with these genes. So yeah, I guess it's a kind of like, this is a blunt hammer approach, and if you're wanting to get specific regulation of certain environmental stresses, more work needs to be done to understand where the signals are coming in and which peptides are being cleaved where, and the proteases and the regulation of the proteases. Yeah, there's many kind of factors that still need to be. But it's clear that speechless as well. I think I was looking at one of the questions in the chat, which is kind of linked, I think, about developmental regulation of rice. So speechless is kind of regulated via epigenetics and like humidity levels. I said about EPF, there's the regulation of CO2. There's also, I think, GWAS studies that continually pull out EPF as kind of water saving kind of like, genes when people are looking at different populations. Yeah, there's a huge amount of potential for manipulating these different toolkits for like regulating, different responses to abiotic stresses. And of course, if you start looking at organ-specific promoters, you can target just one part of the plant as well if you just wanted to target leaves and not other areas, so it's these are quite basic experiments in some ways, and that there's much more defining that needs to be done to really optimize plants for future climate.
Thank you for your answer. Actually, in this chat box, we also have a question from Justina. Please, Justina, if you can open your microphone, you can read your question directly to Dr. Caine. Okay. Wait. Before Justina, Nurfianto also asked a question to you, Dr. Caine. Yes, I will read this question. The first one. How do environmental factors, both climate and microclimate, affect the expression and regulation of stomatal development genes in rice? I think you already mentioned that. And the second question, how is the relationship between stomatal development and the genes that regulate it for fruit and tree plants with water flux parameter, etc.? And the third question, how are the implications of this stomatal research-related condition in Indonesia, where there is the development of hydroponic rice cultivation in greenhouses, and many rice breeding studies are in greenhouses? It's also from Norfianto. Dr. Caine, you can answer the question.
So, as far as I'm aware, there's only limited work in trees, but there is more work, like this kind of technology is being applied in many different like crops and species now. So there's work in potatoes recently that's come out, which shows that like EPF and EPFL9 are positively and negatively regulating development. So, and then we have work in tomato. There's work in grapevine as well, which shows you can reduce tomato density in grapevine to increase water fluxes. I think there's also work in poplar. So people have been trying to increase the number of stomata in poplar so that you can increase biomass accumulation by having higher levels of photosynthesis because you have more stomatal openings. So there is kind of quite a few different crops and things there. This is kind of something that's being looked into. Trees is tricky because obviously the generation time is kind of slower, and it's hard. There's no evidence yet for like how much impact it has on the macro scale with big trees, and that's something that should be really interesting. If there's whole forests that are struggling with water, then the mass reduction in water might be really helpful. Okay, so there's the obviously when you can provide enough water, and things like methane release is not an issue, then you could argue that you can still reduce the total amount of water that's being used by the plants under hydroponic situations, which if water is really important, and you don't have enough even like in a hydroponic situation, or you want to save water, then the kind of transgenic or CRISPR, because the work I showed today was on genetic modification of EPF1 and EPF2 via overexpression, but there's also research that's coming out now with CRISPR-Cas9 knockout of EPFL9, the positive regulator. so it reduces the number of stomata in a kind of non-GM way, gene editing. So you can do the potential is you could use CRISPR to sort of reduce this and then reduce the total amount of water. So, but I think the really kind of the most important uses for this is places where there's not a lot of water, and you might it depends on the total amount of water that's available to you. It might open up areas that are right on the border at the moment of not being able to cultivate certain crops because they don't have quite enough water. But this might be the difference between cultivation and not cultivation, and or using water in different environments. So if you've only got a limited amount of water and you can apply slightly less to your crops, you can have slightly more for other purposes within society. So yeah, the hydroponic thing is if you have got enough water and everything, then it's not necessarily necessary. It's more aimed at like where there's kind of stress, I think is probably, yeah.
Thank you, Dr. Caine. This is also 3 questions for you from Justina. Okay, I will read because Justina has trouble with microphone. The first question, both stomatal density and stomatal aperture determine the potential for CO2 movement as long as water through transpiration drive photosynthesis. In your research, which one is more important for photosynthesis in water deficit conditions? Some stomatal aperture or stomatal density, and why? And the second question, measuring assimilation rate and transpirations is also related to water use efficiency. How do we relate water use efficiency calculation based on biomass to total water consumption and then intrigue water use efficiency calculated from gas exchange data? Okay, the third question, you have a lot of questions here. In relation to my research in rice screening based on the response to the effect of increasing CO2 and temperature, is it possible to get a decreasing rate of stomatal conductance with an increase in temperature while photosynthesis increases? So that's 3 questions addressed to you, Dr. Caine.
Okay, I'll start with 1 and then let's go through number 2 again in a minute, like when one's finished, because I can't hold all that information in my brain. I guess the way to sort of answer this is kind of stomatal. I think stomatal density seems to be kind of a long-term ecological strategy. So, if you know over time periods, like you are going to grow more in a dry environment or more in a wet environment, then it's like investing. You're going to speculate more, or you're going to be more saving your money because you know that you may be going to come across danger soon. So yeah, I would say that if there's not a constraint so much based on water, then plants will be more speculative and have their aperture more open, and therefore like they can conduct more photosynthesis. But yeah, there's a very, very complex question that, because different plants have very different ecological strategies. So some plants have like a risky behavior and some plants don't have a risky behavior. So it's not an easy question to answer that, because it totally depends on the kind of environment. And sometimes it's, it's just by chance that some plants will decide that this is their like most successful strategy for success. And it'll go the other way in a different environment. So I guess the real answer is you can't definitively say one way or the other, and it's linked to the ecology of the situation, like, because it depends on what the plant is facing in its environment naturally. So that's the first one. Okay, so I mean, this is talking about biomass and water use efficiency and total plant water use efficiency. So this is kind of why I think it's important to when you discuss kind of intrinsic water use efficiency or instantaneous water use efficiency, it's important to consider plant size as well, because assimilation and stomatal conductance and transpiration is normally calculated on a unit area basis. But of course, if a plant has more unit area, then it will be magnified in that case. So the measurement at the leaf level is really important for a diagnostic approach, and a first look. But yeah, if you only take that in isolation and you don't consider the whole plant as well, then you may become confused in it. So it's important to, we've done experiments where we've measured the whole amount of water applied to the plant for the whole duration of the plant life, and then we have divided it by the total biomass, and we use that as a value for water use efficiency. And then we also use erga for instantaneous water use efficiency. And then of course you have isotope measurements as well, and you can look at total grain yield versus water use. So the answer to that question is probably that you shouldn't ever take one value as a total kind of explanation of the plant's performance, and that it needs to be considered in relation with biomass, water use efficiency, accumulation, and other factors as well, like grain. How much water does it take per unit grain as well?
Thank you, Dr. Caine. Novianto said, thank you so much for insightful answer, Dr. Caine. Thank you.
Welcome.
So we are waiting for one participant to one more question from participant, because we have like 5 minutes left. Anyone want to ask question or comments? Oh, right. Okay. Oh, okay. We have a question.
Langsung saja gitu ya.
Yeah.
Okay. Yes. Thank you very much. I would like to ask to Professor Umezawa first. Sometimes ago, some years ago, I am one of the subtract member when Professor Umezawa supervised in this project cooperation between subtracts and LIPI. Professor Umezawa, yes.
Okay.
Yeah, sorry. Yes, my question is to Professor Umezawa first. How far the development of biomass energy, especially of lignin content, can substitute to fossil energy in the developed country, or in Japan, or in the world?
We believe
Yes.
so. These are in part. And in Japan, we have a project, and we have established a kind of, association, and it was reorganized to a company. And we are now conducting the project for the social realization of sorghum pellet use. So at least in part, we can replace the coal or other fossil fuel, using sorghum. But we need to solve many, issues.
Oh, yes.
And the sorghum cultivation should be, there should be a beneficial for the local farmers as well. So it's very important. So that means there are many things to be solved for the social realization. Of course.
Okay.
Thank you very much.
Thank you very much. The next question, Prof. Umezawa, because cooperation between subtraps and LIPI last time ago, there is a very important to support researcher and support for some equipment that's for our office. My question is, can the project be continued or cooperation in the next year?
Our project was over just it was over in 2022. So after that, I have not yet visited Indonesia, so I don't know the current status. But for the time being, project has already been over, so
Yes.
we don't have any, big budget to support. But I heard that in your side, Green side, I think you are all also you are still continuing the sorghum project. I think
Yes.
maybe some of the audience know that much better than me.
Yes.
And another Satoru's project is ongoing, I think, regarding the rubber project. But I think some of the researchers are involved in that project. Pakureza, do you know that? Matsui Sensei's Satoru's project from Riken?
I know some researchers from Britain also involved in that project. One of them is Satya.
And Mitani-san is involved in it, right?
Yes, in rubber, the Rubber Research Center. Hi.
Yes.
Thank you. Thank you very much. Boleh nggak satu lagi ke Profesor Caine?
Yeah.
Thank you very much, Prof. Umezawa. Yes. Yes, I have a question to Dr. Caine. Dr. Caine, I'm very surprised with your presentation about the somata from 1 dimension, 2 dimension, and 3 dimension. And for the 3 dimension, you present about how the drone can catch the area and can predict the stomata. I think this is a new technology, especially in Indonesia. I don't know what's the equipment to capture this this vegetation. Can you tell me? Yes.
Yeah. So this work we did was with quite a basic drone, and it was actually 3 or 4 years ago now. So the technology is coming on really fast, even since we made these ortho mosaics. And now there's some kind of DJI. So the drone that we used for the thermal was called an Anafi Parrot, which is kind of very low resolution and not very high sensitivity. And even with that one, which was quite cost effective, you could still get a reasonably good result. But the quality and the sensitivity of drones in terms of the it's called the millikelvins, and the resolution per pixel is getting better, and more cost effective. So the one that we just got a new one, but we haven't flown it yet, but it's probably 3 or 4 times more sensitive and much higher resolution. So it's definitely and also when we did this, the software wasn't very easy to use, but the software that is available to make these ortho mosaics is now getting much easier to make. And making the flight program for the drone when you set it off is much more automated now, getting more simple all the time. So I'm just trying to remember while I'm talking to you what the name of the latest drone is, but I can't remember the model number off the top of my head. I will just have a quick look. But if you have any other questions while I'm looking for this model number, I'm happy to answer that. Just checking now. Won't be. BGI metry.
Mau presentasikan yang itu turun.
So the one that we're going to start using soon is called a DJI M30T, and that is that one's kind of got some new software with it, which makes it a lot easier to operate. So if your institute's looking at kind of Mutiara, who invited me, we've talked a little bit about drones and things, and optical drones and thermal drones. But yeah, this one is a thermal one, which is very good for looking at differences in water profiling. So yeah, it's a M30T DJI. Do you have questions?
Okay. Yes. Thank you, Dr. Caine.
Okay. Thank you.
Thank you so much for the great talk, for insightful presentation. For Okay, sorry, this technical situation. Thank you very much for the presentation, for insightful talk from two speakers. As we have a certificate for our speakers. This will virtually show up in your screen, but we will deliver it through your email. The first one, this is the certificate of appreciation to Professor Emeritus Toshiaki Umezawa. Thank you for the talk. I hope someday we can meet not virtually, but directly in Indonesia or anywhere. Yeah, thank you very much, Professor, for coming to our Botany Talk series today. And the second certificate of appreciation will be given to Dr. Robert Caine. Thank you so much, Dr. Robert Caine. You spare your time in the morning to deliver a presentation to us. I know it's like early in the morning in UK. Thank you so much for the talk. Thank you for the presentation. It's very interesting. I hope you can come to Indonesia in the future. Thank you so much. And for the last, we have also door prizes for 3 active participants. So because we only have like 3 participants to ask a question, the first one is we will give a door prize to Norfianto. To Norfianto Giar Pangidung. Please, can you please send us your contact to us through this platform, and also to Justina Febri Salsina. Also, please give us your contact so we can contact you. And the third one to Professor Wahyu. He is beside me, so I don't need ask contact from him. So thank you very much for we were waiting for your contact to participant Norfianto and Justina. Thank you very much for today. Thank you for all participants who stay here until this afternoon. Thank you, Professor Umezawa. Thank you, Dr. Caine. I will end up the Zoom meeting. We will meet in the next Botany Talk series in the next month. Thank you very much. Good afternoon.
Thank you very much. Terima kasih. Arigatou gozaimasu. Terima kasih. Selamat semuanya. Akhir.